Glycolysis starts off with glucose. 2 ATP are added, and it becomes a 6-carbon diphosphate sugar. 2 ADP come out of this reaction. With the help of the enzyme isomerase, it becomes 2 3-carbon isomers. 2 NAD plus go into this reaction and become NADH, allowing room for the bonding of a phosphate group. 2 phosphate groups then come in and bind to these isomers. Four ADP are then added and 4 ATP come out with a product of water. It then becomes pyruvate.
The pyruvate then go through, are actively transported through transport proteins in the mitochondrial membrane, outer membrane, and then 2 NAD pluses are reduced to become NADH, and then also 2 carbon dioxides are released and just diffuse out of the membrane, and 2 coenzyme A's bind to the pyruvates, becoming acetyl coenzyme A.
The acetyl-CoA carries the pyruvate to the citric acid cycle, then leaves. The pyruvate bonds to 4 carbons and begins its cycle. During this cycle, through a series of oxidation reduction reactions, 2, 3 NADHs are produced, 2 CO2s are produced, and 1 FADH2 is produced. Also, a phosphate group is added to ATP and produces ATP. This entire cycle is completed twice for each glucose molecule, once for each pyruvate.
In the next stage of cellular respiration, the electron chain uses the ADHs and the FADH2s in these proteins that pump H pluses to the inside of the cristia. This concentrated H plus powers ATP synthesis, which comes back across and produces ATP using the ADP in 1 phosphorus group. This can normally produce 28 to 30 ATPs. The electrons are passed through each protein, which, as it goes along, each protein is increasingly electronegative. They're moved by carrier proteins that move between the big proteins.
So with fermentation, it's basically done all at the substrate level phosphorylation to produce 2 ATP per glycolysis. It's done without the presence of oxygen, and we'll get started with alcohol fermentation. This happens in unicellular organisms like yeast and bacteria, and it basically has glucose undergo glycolysis, where 2 ATP are produced, and 2 NAD pluses are reduced along with producing 2 hydrogen ions. The product of this reaction is 2 pyruvates, which then undergo a series of reactions that release 2 carbon dioxide and become 2 acetaldehydes, which then go and are reduced from the NADHs So basically the NADs can drop off their H's here, and this becomes 2-ethanol, which is an alcohol. The NADs basically just go around, drop off their electrons, and then go collect more. That's alcohol fermentation. So lactic acid fermentation is really similar to alcohol fermentation in that it uses substrate level phosphorylation to break down the glucose through glycolysis, producing 2 ATP per glucose molecule. 2 NAD pluses are reduced to 2 NADHs, also releasing 2 hydrogen ions, and producing 2 pyruvate, 3 carbon sugars. It is different in the sense that these pyruvates are reduced directly from the NADHs to become 2-lactate.
The pyruvates bond.
So for the next stage of what are we doing? These power proton pumps. Darn it. We go to the electron chain. The NADHs. Into the concentrated cristae of the mitochondria. These allow for it's going to be in the video.